Thank you for reminding me. Okay. We already went over the syllabus. Okay. So I guess the goals for this lecture, so you guys are aware, is this is going to be a diagram we're going to see quite a bit throughout this course. You can break down all instruments, like we literally have our black box here. And you can break down every single instrument to what's the source of whatever radiation or probing type interaction is being sent to the sample. The sample then interacts with whatever physical variable is coming from the source, and then we detect it somehow. So today we're really focusing on what our sample is, what types of samples do we care about, and we have to review, I guess, what aspects of biology we want to study or use our instrumentation with. And for that, we need to understand the terms that are used in biology. And the organization. So we're going to be going over like how we categorize living things. Yes, the organization in terms of cells, organelles, what biomolecules, if we're getting down to the chemistry scale, play a role, and also some of the central dogma of biology. So review for some of you guys, refresher from a couple of years back for others of you. And before we get into, I guess, these terms in organization, I do want to point out a few things. In terms of biophysics, I would say many physicists are might be put off by biophysics at the start or find it hard to understand because biophysics is filled with jargon. I just want you to recognize that and don't be scared of it. And a lot of this jargon is historical. For example, I've been writing some grants based on this protein called SMAD2, and I was like, okay, what's SMAD? What does this stand for? And like when you actually track down what it means, okay, this actually has a physical meaning, a signaling protein. It's a protein that induces a signal that changes something in the cell. But this part here, this mothers against decapentaplegic homolog, I mean, that was from a joke from some grad student somewhere where they were making a joke about like it's similar to mothers against drunk driving. So that doesn't have any physical meaning at all. So, and then there's multiples of these proteins, so that's why it has a 2. But realize like, then all these proteins were called SMAD because of that joke that they identified, and they didn't know this protein would be so important. It can also be related to some physical observations. So there's also this signaling pathway called the hedgehog pathway, where this HH is the hedgehog protein. And this was because it was first studied in fruit flies, and they saw if they knocked out the gene for, if they removed the gene for this protein, the embryos for these fruit flies would have a hedgehog type pathway. So then related to that, then they find another protein related to this, and then they call it Sonic the Hedgehog, like the video game. So there's also like the hippo pathway, because removing this protein hippo would make the cell tissues overgrow, looking like a hippo or so. So don't be intimidated by these terms. You really have to look at, if you're getting into something as involved as a signaling pathway like this, you kind of have to identify the first time you're looking at it, okay, who are all the players? They have these weird abbreviations, but you don't need to know like all this background. So don't be too intimidated with that. Don't let it scare you away. Also, before we get into the biology, we need a reminder of some of the chemistry as well. We're going to be using organic chemistry notation for some of the chemicals and the like. So if any of you guys want to call out, I guess, with this ring structure here, any of the chemists here, or can you tell me what this structure is or what chemical group this is? Or in the chat.
Benzene.
Yeah, so this is our benzene, and these two lines represent a double molecule or double carbon bond. Each of these vertices here, where I'm pointing with the orange arrow, is going to be a carbon atom. We can also say this is conjugated, where you have alternating single and double bonds. That's what we mean by conjugated. Okay. With this, where I'm pointing with the green arrow, can anyone tell me what these dash lines represent?
Like a bond going into the page.
Yeah, so that's telling us it's going backwards. It's telling us something about the 3D orientation. So for the physicists in the class, this would be similar to drawing a vector that's going into the page, like with this notation. While this solid line, or I guess the solid triangle here, would be something coming out of the page as well. So a vector that we would draw like this in physics. Here again, this is going to be a carbon atom here, and it's bonded to 4 different functional group, or 4 different groups here. So this is going to be a chiral center that has a orientation dependence. And then also important chemical groups here, we have an amine. And then this group here is a carboxylic acid, and these are important in biology. There's lots of different functional groups. I'm not going to go over all of organic chemistry, but just for the physicists in class, a reminder of this chemical notation. So with that recap of, let's see, is there anything else? Yeah. And then also I want to note that not all hydrogens are drawn in this notation. So here you can see this carbon at this location has two bonds here, 1 bond here, and then there's also a hydrogen that's not explicitly drawn in this organic chemistry notation. So with that recap of some chemistry, biology, we can get into some of the other topics. One being that we're going to start large here and talk about the organization of life. That living thing, we can say an organism, those would be on the scale of 1 meter or so, and organisms are organized into eukaryotes, so that would be animals, fungi, and plants, and they can be multi or single cellular. Then there's also prokaryotes, which would then be bacteria, and archaea, which we won't go into archaea that much, but bacteria we will be talking about. So these are going to be single cell, and they have no nucleus, which we'll talk about, while eukaryotes have a nucleus to store their genetic information. Then the next level of organization would be organs, which would be on the order of 0.1 meters. And these are going to be grouped tissues or cells that carry out a function, a specific function. In the organism. While tissues are going to be on the order of millimeters or centimeters. And these are going to be cells grouped in 1 region of space to carry out a specific function again. Then going even smaller, then we have our cells, which are going to be our fundamental unit of life, that are going to be like 10 microns to 100 microns. This is a 4. And this is also used as a common model organism. If you're working with E. coli, for yeast, or a model for, I guess, biofilms on the prokaryotic side. Then also you can think of like HeLa cells, CHO cells for model, I guess, human samples. And also worth noting, viruses, those are going to be more on the order of tens of nanometers. And they do not have the ability to reproduce independently. So people debate if they're living or not. I err on the side of not living. So those are the different scales, and we're going to cover all of these scales throughout this course. We're going to be starting on the smaller scale to start, and then throughout the semester, we're going to move up to larger. It's also worth noting this terminology of in vivo, where people use that to indicate that they're doing a study within an organism, versus in vitro, where that would be a smaller substituent of an organism. I would say within cells, people will still use the terminology in vivo if they're single cell organisms. That's contested based on whatever field you're working in. And then also it's worth noting that we're going to be doing a lot of in silico techniques as well, which means in glass. So that's even outside of a cell or these living organisms. Okay. So getting into more detail on the cell, I said before, this is going to be our fundamental unit, if I can spell, of life. And they're considered that because they're functionally autonomous. And they can reproduce on their own. Because they have all the biomolecules needed to replicate. We already went over that there's the prokaryotes and the eukaryotes and their classification. It's also worth noting that you may come across work focusing on the cell cycle, that the state of the cell will depend if it's growing, if it's dividing, so mitosis being the division, prepping for division as well, while interphase is the growth where the cell is not dividing. So we're not going to get into all the details there, but be aware that the dynamics, differentiation, growth, motility of cells are all, those will vary individually based on where they are in the cell cycle. So then getting into more details about subcellular organization, getting down to organelles, this is going to be more on the order of, I guess you can say, microns to nanometers based on the size. Organelles can be membrane bound, enclosed in a membrane, or they can be membraneless. And this is an extremely hot area of biophysics right now, are membraneless organelles. And this really, for the people who are in macromolecular sciences, this comes from ideas of phase separation and ideas of polymer physics. So I'll bring this up later. It also has to do with intrinsically disordered proteins that have more, I guess, polymer random coil structures as well. External to all these organelles is also the cytoplasm. So that's going to be your, and there's also the nucleoplasm within the nucleus. This is going to be your cellular solvent. It's not just water. And be aware that the cytoplasm isn't just, some type of buffer or water. It is extremely crowded. And it's concentrated with a lot of proteins and other biomolecules and structures present. So I'm going to go over really quick some of these organelles. One of the questions in the first problem set is picking one of these organelles and going into more detail. They all have their own organization and structures within themselves as well. So I mean, the nucleus is where we have genetic material stored. It's going to be around 1 to 10 microns in size. It's going to be the largest organelle. And it has, you can see here, they're even emphasizing that transport through the nucleus is through pore-like structures that people have studied. And it has the bulk of the genetic material. And we're going to talk about this nucleolus, where the chromosomes are located at, and how DNA is packaged within there. After the nucleus, we also have the ribosome, where RNA is translated into protein. So this is where RNA to proteins. It has subunits and its own RNA within its structure. There's many ribosomes within a mammalian cell, greater than 10 to the 7th. So you can see all these little red dots in this cartoon here. Proteins are assembled in the endoplasmic reticulum. You can already see that it's noting the different areas of the ER, the smooth and rough structure. We have the Golgi that packages proteins. We have the mitochondria, everybody's favorite organelle, the powerhouse of the cell. And it's worth noting it's studied quite a bit, where it has its own DNA within there. It has a double membrane. It's a very unique organelle. I'm just going to put ATP, the energy unit within the cell, to indicate that it has energy. We have the lysosome that breaks down waste. Similarly, the peroxisome with hydrogen peroxide. There's energy storage as well with, let's say, vacuoles in animals and fungi, or starch granules in plants. Chloroplasts are within plants, and then also it's worth noting we have the centrosome and microfilaments, that there's a cytoskeleton within these cells that's for structural support. And the cytoskeleton also plays a role at interfacing between the cell membrane and the extracellular matrix, or the environment around the cell as well. So you'll hear with the cytoskeleton, we have microfilaments here. You'll see microtubules, actin, intermediate filaments. There's different chemical constituents with that. I just mentioned here we have an illustration, I guess, of the microfilaments of the cytoskeleton. An important organelle, you can call it itself, is the cell membrane. And in very simple models of thinking about the cell membrane, we're going to be talking about lipids later. You'll have this phospholipid bilayer, where you have hydrophilic heads, hydrophobic fatty acid tails that form this bilayer. And a lot of pictures of the cell membrane are, I guess, simple. And this isn't how the real cell membrane is. It actually has many different proteins. It has linkages to inside the cell and then outside the cell with the extracellular matrix. There's transmembrane proteins here, where this protein can feel forces or exert forces on the surroundings. It's, again, very crowded, confined environment. You also have carbohydrate modifications that change the recognition or the phenotype of the cell on the outside. Molecules like cholesterol or the different types of lipids that are involved, you can have gel-like phases or liquid-type phases. And that the differences between, I guess, gel and liquid phases, you can have solid, the gel regions might be more solid versus liquid regions that can move around. So this leads to the hypothesis of lipid rafts that you might hear about the cell membrane. So there's a lot of details we could go into with the cell membrane, and we'll, as we talk about studies that look at cell surfaces, we'll talk about that. And it's also worth noting the function that the cell membrane has of being a barrier, identifying what's the cell versus what's not the cell. And it plays a very important role in like molecular transport, signal recognition, osmosis, or any of the pressures or the amount of water that's available in the cell. And it's also a dielectric capacitor. If you think about the differences between ions on either side of the membrane. So those are some of the concepts of the cell membrane. So we've been working at the top, working our way down to the cell, within the cell, the organelles. Now let's get at the small molecules that make biology work. The important thing is that carbon is what you'll see the most commonly. Carbon is a key organic element for you. The difference between organic and inorganic chemistry. And this is a key atom in life because it's a group 4 element. It can make 4 different bonds. It has intermediate stability. And what I mean by that is that it's stable enough to carry out functions without undergoing a reaction or a change, but its modifications are low enough energy that they can be easily chemically modified. To change the function. So that's why you see carbon so commonly within biology. There's also a lot of other small molecules that are key in biology, including most commonly water. Can't cover everything about water, but we can call it the universal biological solvent. That's because it has unique dipole properties with hydrogen bonding. You guys all should have seen this in Gen Chem. Plus, this is supposed to be a minus here. It has unique orientation properties. You can spend your entire career trying just to understand water, so we won't go over all those details. Ions you'll also commonly see in biology that play a role in signaling, in pH or electrical gradients or potentials, or regulating protein function. And these include sodium, chloride, carbonate, potassium, you'll hear potassium pumps, magnesium, iron, like with hemoglobin and red blood cells, zinc, with zinc fingers on proteins, and of course the hydrogen ion as well. And then of course the energy unit within biology is ATP, adenosine triphosphate. And this has the ability of undergoing hydrolysis to get to ADP to release energy. So going from ATP to ADP, this means it has 2 phosphates instead of 3. That releases about 18 kBT of energy. So this is a unit we're going to be using, kBT, the thermal energy unit for biology. So this is about 18 kBT. When I was talking about carbon up here, to make a change, it's around 100 kBT. So you can see with just 5 or 6 or so ATP, you can undergo those chemical changes to modify carbon, single carbon bonds. So those are the small molecules, the atoms that you should be aware of in biology. And then let's get into some of the macromolecules. Since you're just talking about the cell membrane, let's talk about lipids, or we also call them fatty acids. Where the important thing here is that you have This is our hydrophilic group here based on glycerol. This glycerol is shown simply here, but it can have like phosphate groups or amine groups that can change the charge. So the ability to have different chemical groups here allow these to have different properties. The fact that you'll see phosphocholenes, and those are groups that have both a positive and a negative charge. So those are called zwitterionic head groups on lipids, and those are typically found on cell membranes because they resist protein absorption. So the reason that you don't have all this protein from the bloodstream or the surroundings coming and absorbing onto the cell surface is because of those groups. The glycerol group is then linked to the hydrophobic chains shown here. So that's going to be our fat. That's where the fatty acid comes in, the fact that it's hydrophobic. These hydrophobic chains you can see here, they can either be saturated or unsaturated. And what this means is here, since there's a double bond, that means it doesn't have the maximum number of hydrogen atoms that could be on that chain. So based on the saturation, you'll have different temperatures that these lipids are either in the gel state or the liquid state. So the fact that we have the hydrophobic and the hydrophilic portions means that these molecules are amphiphilic, if I can spell, which means it has both phobic and philic portions. And this property allows them to self-assemble, where I've shown different ways that fatty acids can self-assemble. A simple sphere with one layer would be a micelle. We've talked about the bilayer sheet that is sometimes used as a model of the membrane, very simple model that doesn't have all those proteins. Or it can self-assemble into a spherical shape with the phospholipid bilayer as well, and we call those liposomes. And the amphiphilic part of this also allows for like a large potential to pass through a water molecule through these layers. So it's also not energetically favorable for a water molecule to pass through. So the energy for passing a water molecule through a bilayer sheet, the free energy is around 65 kBt. If we plug this into the Boltzmann equation where we put exponential over delta G, RT, this will result in a probability that you would see a water molecule just spontaneously pass through a bilayer sheet is 10 to the negative, up to the negative 28. So that's showing you just even for a simple small molecule of water that it can't pass through this tight bilayer sheet, showing how secure the membrane is for keeping the cell secure, keeping those molecules within the cell. So different, that's why the membrane has to have different transmembrane proteins or channels to pass these small molecules through. So based on this description that I've provided of different properties with the lipids, I guess a quick, let's see if I can do a Zoom survey, or we can just put it in the chat. Add a poll question. Let's see if this works. Did I make this crash? Okay. I don't know how to use the polls well enough. Can you guys still see the PowerPoint slide?
Yeah.
Yes.
I guess if you guys want to add in the chat what letter you think this is, which molecule would be unsaturated, zwitterionic, and amphiphilic. Okay. Yep. Looks like most of you guys got this. Yeah, it's B that we have. Where'd my pen go? That we have the zwitterionic group here. Go back. Phosphocholine, the glycerol group. The fact that this is unsaturated with the fatty acid makes it amphiphilic. Also within these examples that I've shown here, this chloride ion would diffuse away. That's just how it's sold to have charge balancing here. And then this molecule here is cholesterol, which doesn't look similar to these fatty acids, but is considered a fatty acid since it's found in membranes and can, again, change the liquid versus gel state of the membrane as well. Okay, so moving on to proteins, some time we have left. Amino acids are going to be your small molecule building blocks for proteins. And the important things with the structure is they have carboxylic acid and an amine group on every single one. You can see that here and here. And those two groups then connect through a peptide bond so you can create strings of amino acids. You can see that there's a variety of natural amino acids. There's 23 natural amino acids. This table that you'll see quite frequently if people are working with proteins, it shows 21, and those are the ones that can be naturally produced in the human body. And you can categorize these amino acids based on, you can see here that we have positively charged or negatively charged groups, polarity, hydrophobic groups, so they can be classified on the different properties that they have. I also want to note that the names of the amino acids, I'm not going to make you memorize these like a biochemistry class. You can see that we have the name arginine here. They also have a 3-letter code, arg, and then they have letters, single letters to represent each of these amino acids. If you're looking in the literature at some study where they've mutated proteins, you might see some notation where it's like, let's say, R23L. So that means that the protein originally had an arginine amino acid at the 23rd position in the primary sequence. We'll talk about that on the next slide. And then it was mutated to a leucine. So that's how these single letters you'll see commonly in the literature. It's also worth noting with these different amino acids that the charged amino acids can change based on pH. They could either be neutral or they could be charged. In the special cases here, you can see that cysteine has a thiol group or a sulfur atom, and this is used commonly to functionalize proteins with specific reactions, or these also form disulfide bridges. They'll interact with another cysteine molecule. These amino acids down here, tyrosine, tryptophan, and phenylalanine, you can see that they're conjugated, that they have those benzene groups with repeating single and double bonds, and those are taken advantage of for optical measurements. And then the histidine with its charge here is used in protein separation. So these are all techniques that we're going to be talking about throughout the semester, and we'll those will come up in those lectures. Also worth noting that there's unnatural amino acids that people have engineered, so they can have very special chemical groups like quaternary amine that can undergo click chemistry. So people, that's more on the bioengineering side of proteins. There's peptide synthesizers that can make short polymer chains of these peptides, of the amino acids. And then that these amino acids are chiral, and they're naturally L-chirality. And that will come up in, I guess, 3 lectures or so. So amino acids make up proteins. You can say it's kind of an arbitrary definition, like a short chain of amino acids is a peptide that's going to be less than 50 amino acids, while proteins would be, I guess, arbitrarily greater than 50 amino acids. They can vary greatly in size. Insulin is around 53 amino acids, while a large muscle cell protein, titin, is around 30,000 amino acids, so they can have huge differences in sizes. There's different degrees of structure within a protein that you should be aware of. Primary is just the order of the amino acids. Secondary is going to be hydrogen bonding forms between the amino acids, forms either an alpha helix, a beta sheet, or it can remain a random coil. So I've shown that over here. You can see that the hydrogen bonding between the amine and carboxylic acid groups, or I guess the ketones here, can either form these pleated sheets or the alpha helix, or here this is showing the random coil, which you'll also see as an intrinsically disordered protein. It just can take a random configuration, like a random polymer coil. Tertiary structure is going to be the orientation of the beta and alpha helices relative to one another. This is going to be driven by van der Waals forces. Again, hydrogen bonding, hydrophobicity. A note with hydrophobicity, with proteins, they have a hydrophobic core. Let's see. It's a new chat. So you can imagine these proteins are going to be in an aqueous environment. So in the center, it's going to be more hydrophobic. Amino acids in the center, more hydrophilic or charged on the surface. Quaternary structure is the assembly of subunits or monomers. So you can have a larger complex of multiple proteins. And then there's also quinary structure, which you may not have heard of, but quinary structure is surface interactions between proteins. Assembled proteins that are driven by crowding, charge. So these are intermittent and short-lived, but they can last longer depending on, I guess, the osmotic state of the cell environment, the surface of the protein, and based on charge, it's also worth noting this terminology of Pi, which is the isoelectric point of a protein, and that's where the protein is neutral at a certain pH. So just as a quick illustration here of the primary sequence, here you can see those 3-letter codes for the amino acids are 1-letter code. That's how you'll see the primary sequence. Secondary sequences, those alpha helices and beta strands, but they can still rotate freely. And then as the protein folds and begins to orient itself, those strands and helices are in domains. And finally, you can see that there's different subunits here that then can come together for that quaternary structure. So then the assembled quaternary structure coming into contact with other proteins would be the quinary structure. And there's a lot of slides here on proteins, but that's because they are the workhorses of the cell. The question in the chat is, are disulfide bonds between cysteine residues primarily involved in tertiary or quaternary structure? Also, what level of protein structure is pi stacking involved? So disulfide bonds you'll see show up in quaternary or in tertiary structure, I would say, and also quaternary structure when subunits are coming together. So I'll say that they contribute to both. And pi stacking also be involved in tertiary structure. And when we get into the spectroscopy measurements of protein, we can talk about the pi stacking as well. But I would say you'll see hydrogen bonding and hydrophobic forces coming more into play than pi-pi stacking. Pi-pi stacking, disulfide bonds, those are going to be in very specific areas of the protein, while the hydrogen bonding and hydrophobic forces you'll see everywhere within the protein. So getting back into the function, workhorses of the cell, I'll write that out. And they're about 20% of the mass of the cell. And their functions are derived from their structure. And this is discussed a lot with proteins. Structure is related to function. So people are really studying protein folding because this random string of amino acids, how does it always go into the same structure so it can carry out this function? So we're going to be talking about the techniques that look at protein folding, where they're either looking at the structure at very high resolution statically, or they're looking at these changes over time and looking at temporal information as well. Common protein functions, you'll see enzymes, where they're the biological catalysts, where you'll see examples of ligands or small molecules coming into a pocket within a protein. That protein undergoes some conformational change and brings these molecules together, undergoes some type of reaction, and you'll see the terminology lock and key, which is very historical. I want you to understand that that's a little bit outdated. Some enzymes function with lock and key mechanisms where these ligands come in and really lock in, but there's many other small molecules that can come in and interact. It's not just a specific ligand every single time. Also with enzymes, you'll hear about allostery, and that's when a molecule comes in at a different site away from this active site and causes a conformational change within the protein. Other functions are motor proteins, where you go from, I guess, energy to work, some type of mechanical work. You'll also see signaling proteins at the membrane surface. Those are just some of the functions that you'll see brought up. Okay. So I want to go quickly through these last sections of small molecules of sugars and carbohydrates, and these are cyclohexane-based small molecules that then polymerize. So there's going to be monosaccharides, monomers, polysaccharides as a polymer chain, which is shown here for cellulose. And carbohydrates are also chiral with a D configuration as opposed to the L configuration with amino acids. And they play a role in structure, like cellulose in plants, or energy storage, such as starches or glycogen. A big area of biophysics, and when we get into mass spectrometry, is glycosylation. Can't spell. And this is a modification of proteins with carbohydrates and sugars after they're expressed. So each of these colored blocks or structures here represents a different type of sugar group. And then these are modified in the endoplasmic reticulum to have different carbohydrates. And that changes the functional role of those proteins or where they're sent. And this glycosylation or this post-translational modification distinguishes the difference between the number of genes that we have when we shift to nucleic acids versus the number of proteins in our body. So the genes are stored in information with nucleic acids, where we have DNA and RNA, where these just have different sugar units, deoxyribose versus ribose. And the sugar units you can see here with the phosphate backbone that's negatively charged and hydrophilic. And then these pair up with the nucleotide bases, where you have your thymine and adenine pairing up, or guanine and cytosine, while in RNA, this is replaced by uracil, so different base pair there. The different ends, you should note that if it ends in the phosphate group, it's a 5' end. If it ends in the sugar, it's a 3' end. And that most DNAs is B DNA structure with the double helix shape with spacing of 3.4 angstroms between the nucleotide-based steps, but there's also a more condensed form, an aDNA, or a different chiral form in zDNA. DNA within the cell is assembled. It's not just a random double helix strand. It's actually wrapped around proteins called histones that then form into complexes, coils up, and you'll find these stored in the nucleus as a chromosome. We have about 19,000 genes that are coded by promoter regions, start codons, specific sequences of these nucleic acids. But the majority of DNA, 98%, is actually not coding. We know we're running out of time. So I also just want to mention with RNA, there's different forms. Messenger RNA goes between the nucleus and the ribosome. RNA is typically single-stranded, and it can form base pairing within itself. So you can see these hairpin structures within. You'll also hear the terms aptamers, which are single-stranded RNA for sensing applications. tRNA is between mRNA and the amino acid in protein assembly, and rRNA is part of the ribosome. And these all play a role in the central dogma of biology, which has to do with how we go from DNA, our precious genetic code for all of the proteins that stays protected within the nucleus, to RNA that is able to leave the nucleus, which then goes to the ribosome, and it is translated into the proteins that then are the workhorses of the cell. And I also note here other processes, replication, reverse transcription. We can have an entire class just devoted to the central dogma of biology, but we're not going to get into those details in this class, but you should be aware of these different players here. And then finally, just some other things to note that will be brought up. We're not going to be talking about like Krebs cycle or Calvin cycle, but just how different biomolecules are produced and energy. Later in the semester, we'll get into evolution and selective pressures, noting that phenotype, the physical experience, expression within an organism may be different than the types of genes. That's the difference between different cell types. They all have the same types of genes, but they have different expression of those genes, that different distributions of proteins that lead to different cell types. And then later in the semester, when we get to mass spec, we're going to be talking about the omics revolution. And that's a collection of large amounts of data related to genes, related to genetic changes, related to proteins, related to carbohydrates and nutrients as well. So just be aware of that term when we use omics. So with that, that's what I want to cover. Sorry, it went a little bit long. This chapter is uploaded on Canvas. That covers everything we talked about in more detail. And if you really like this topic, you can take entire classes devoted to it. So yeah. Sorry, 1 minute over. Does anyone have questions?
Yes, so are you going to post like readings ahead of class time regularly, or like
Yeah,
this?
so I've uploaded, before this class, that chapter was posted, these notes were posted, typed out, as opposed to me writing them, if you guys prefer to use that and go along with the what I'm writing. The readings aren't mandatory, but if you need, I'm providing that as an additional resource. So since this is an upper level class, I'm leaving it up to you guys to decide how much you need to read or get into those details, especially since you guys have so many different backgrounds and courses you took.
Professor Kifley?
Yeah.
I have a quick question about glycosylation. How much do we need to know about that specifically?
Just be aware of the term that it exists right now, and it's how those sugars are being connected to protein. So once we get into the techniques, we'll talk about like, okay, this mass spec is being used in this way to detect the different sugars and different that are connected to the proteins or cell surfaces and stuff like that. So I just wanted to mention it because that is something that's not really covered in high school biology or intro biology. So a lot of this is just like, here's some of the terms you might hear. So when we talk about them later, or you see them in a problem set, you can be like, oh yeah, I remember that was mentioned. You know, the at least starting point of where to look at that in detail.
Thank you.
Okay. If there's not anything else, have a good rest of your day, and I'll see you guys on Thursday. Thanks for calling in.